our agenda for today is that first we're going to go over the Kaggle competition. Next is going to be what is EEG, what is motor imagery. After that, we'll go over what is actually inside the data set, and then the experiment that it's on. We'll talk a little bit about frequency domain concepts, including PSDs and spectrograms. And then finally, what is needed to follow along, and a walkthrough with a notebook. If we have any extra time at the end, we will get the data set onto everyone's computers, and we can see where it goes from there. All right. Next slide. Hello?
Oh.
Yeah,
all right.
Okay.
Sorry.
Kaggle competition. The Kaggle competition is held collaboratively by DS3 and NTX. We will have gift card prizes for the top competitors, and this will last through week 3 of next quarter. The competition page and rules are up there on the slide, or link to the competition rules are up there on the slide. And any updates will be through our Discord, that we have just set up. That's the competition link. And then for the Discord, will be right there. We will release any information and any updates through our Discord like that. And then you can follow along with the workshop series. We'll be having several workshops throughout the rest of the quarter. We will learn how to extract EEG features, learn about machine learning, and all of it to apply to the competition data set. Right. And I can pass on to Simon.
So since this competition and most of the work we do is on EEG, I think the first question you might ask is, what is EEG and how useful is it at picking up the neural activities? So let's first look at in the case of this neuron right here. Let's say it's firing at those intervals. So you can see those spikes are called depolarizations. And if it's below this regular line, you can see it briefly hyperpolarizes. So in the case of individual neurons, higher means depolarization and lower means hyperpolarization. And if you see a spike here, that's called an action potential. But most of the times, we aren't able to actually inject an electrode precisely inside of a neuron. Most of the time, we're recording outside of a neuron. As you can see, this is an extracellular action potential graph. One thing you might notice is that it appears those spikes are in the opposite direction. This actually makes sense because when a neuron fires, iron flows through the membrane. And if an iron is flowing into the cell membrane, then the same ions are flowing out of the extracellular space. So you can see that the voltages are obviously in the opposite direction. And local field potential, this LFP, is also a terminology many people use. It's basically a summation of many of those extracellular action potentials. And you can still see like over here, those lowest points in this graph correspond pretty well to the firing of individual neurons. As we take one more step back, we can see the techniques scientists use the most are ECOG and EEG. Those two are actually very similar. ECOG is basically an EEG that is sitting inside of the skull. And you can see, compared to the LFP, ECOG reflects pretty well of the general peaks and troughs of the individual LFPs. Of course, there are summations and cancellations that eventually goes towards ECOG. But you can see that in terms of the general shape, it reflects pretty well overall. Now, once we move to the EEG, interestingly, we have a lot more noises and way less signals. As you can see, it doesn't have the same amplitude as the ECOG, which is sitting inside of the skull. This is due to the fact that as electrical signals travel through the skull, it gets attenuated, meaning different signals get interfered and noise is being introduced. Because of that, it is best at picking up neurons that are parallel to each other and that are firing in synchrony, facing toward the skull. So EEG is really best at recording neurons that is near the skull and has this layered structure that makes them all parallel to each other, especially like this pyramidal cell over here. Those cells are really structured and they project toward the skull. And part of EEG to detect signals from deep brain structures that are less layered. So that might include basal ganglia and those more deeper brain structure, the deeper brain structure that are not as layered as the cortex. Next slide, please. So now that we know what the EEGs what kind of signal the EEG are picking up, how do we use that information to gain insight into how the electric signals correspond to motor activities? Well, it turns out those neurons in motor, in the sensory motor regions, which is in the picture on the lower right. You can see the red and purple region. We're primarily focusing on the primary motor cortex, and within this region, the neurons tend to fire at 9 to 11 hertz, which is referred to as mu rhythm. This, rhythms in this range also are present in the visual cortex under a different name, alpha rhythm. But when we're referring to motor activities, we generally use the name mu rhythms instead. So how do we see the changes in the rhythms? Well, so our brain has this property that it tends to correspond to the activities in the opposite side of the body. So your right brain generally controls your left side of the body, and your left brain controls the right side. So when you're sitting relaxed, the mu rhythms are present on both sides, both hemisphere of the primary motor cortex. If you move your left hand, that desynchronizes, that disrupts the mu rhythm on the contralateral side, which is like the opposite half of the brain. So using this information, we can theoretically tell if the person is moving their left or right hand just by seeing if the rhythm is present on the contralateral side of the brain. And also, it's not just limited to the actual movements. It is experimentally found that even when you're imagining moving your left or right hand, this property still holds. Next slide. So now that we know how the electrical activity can be picked up by the EEG and how that might help us gain insight to the activities, we need to design experiment that would allow this structured, that would produce a structured form of data that can be analyzed. So a typical experiment that for this kind of study is through placing electrodes near that region. Um, so in this case, on the right, you can see a head with lots of dots. Now that is a form of the 1020 format. In this standardized format, every single electrode has its place and has its corresponding name. So when you see over here C3, CZ, and C4 electrodes, those are always going to be over here, corresponding to those three regions. So that tells you where the electrodes are being placed. Now, how is the actual experiment look like? Well, we have in the case of the BCI challenge data, they had a session have multiple rounds for a single session, and inside of each round, many trials, and each trial lasts for 7 to 8 seconds. It begins with a beep, and in the first 2 seconds, you're gonna see a fixation cross on the screen that tells you to basically focus your eye onto the screen. And sorry, I don't have a picture showing, but there's a cue, either to the left or to the right. That basically tells the subject to imagine moving their left versus right hand. Now the cue is shown for about 1 second, a little bit more than 1 second, and it is assumed that 1 second after the cue is shown, the subject probably is starting to imagine the corresponding motor movement. And that's gonna last for 3 seconds. And then they have a break. That constitutes a single trial. Usually within a single session, there are multiple trials for either left or right hand. So all that data is being recorded and stored in this table, in the form of this table. For different specific studies, they might look a little different, but for our data set, specifically, we have, when you open up the data set, you're gonna see this. So you can see there's time on the most axis. It's always incrementing millisecond interval because the EG is at 250 hertz. And you can see the electrodes being placed here. Now there are also EOGs that are used for filtering out the eye movement artifacts. But it's really the left three columns of data that we actually use. To the right side, you see event start. That corresponds to the start of the motor imagery period. So the unit of the data is so when you first receive the data set, it's gonna be in the unit of, I believe, microvolts. So you need to process it before you can get the data in hertz, which Jillian's gonna cover pretty soon.
Yeah. Okay, yeah, before we jump into that, we actually have a bit of an announcement from the DS3 team about their workshops. So just more workshops you can go to for learning how to extract data from neural data. So I'll let Tanvir present. Oh, I don't think your mic is working again.
Yeah, you're not.
Can you hear me now? Sorry about that. Okay, so my name's Tanvir. I'm the machine learning workshops lead from DS3. And as you guys know, this CAG competition that we're putting together is a collaboration between both orgs here. And with that, we are going to be hosting, so Neurotech obviously is going to be hosting a lot of really useful workshops on feature extraction and how you guys actually like look at the data from a neurotech lens, right? But if you guys want to learn more about machine learning or different other data science methods you guys can use to create your models for this competition, we do also have 3 workshops coming up. We have an intro to deep learning workshop in week 5. And also in week 5, we also have an intro to sklearn workshop. So both these workshops are designed to be beginner friendly and also will prove to be pretty useful for you guys who are trying to make machine learning models for the Kaggle competition. As well as that, we have an introduction to transfer learning workshop the week after that. And what transfer learning is, it's the concept of being able to use like really powerful and pre-trained models that are created by people at Google and other research institutes, and be able to use their previously trained models to get high performance on machine learning tasks in a very small amount of code. So all 3 of these workshops are pretty useful, and so I just saw a question. The DS3 workshops will not be applied to the neural data sets, but with the right, like it'll be up to you guys to write the code on your own. But these concepts will be very easily be able to be translated to your guys' own projects for this Kaggle competition. So if you guys have any questions about that, feel free to like message me after this or anything like that.
Cool. Thanks, Tanvir. All right, so just continuing on with our slides, we have some signal processing that we can do on the neural data. So the raw data is in voltage form, where this can be represented in the time domain here on the left. You can see that when we have different voltages, we have different heights of this graph, and over time we sort of get this like really messy squiggly line. And in order to sort of understand this in a more, I guess, stationary way, we can sort of put this through in the Fourier transform and get a frequency domain out of it. And so here on the left side, on the left image, we can see that once we put it through the FT transform, we get a frequency domain where version of the output where you can see how a spike sort of happens at certain large frequency, like it's like the amplitude of the sinusoid that we decompose the original time series signal into. So we can get representations of the power of each hertz, pretty much. And so this is kind of relating back to the question that Michael had earlier, is how do we get like a hertz format of this data? And so this is kind of how we do it. On the right-hand side, we can see an image of a bunch of simple waveforms, and then the FFT of those simple waveforms. The most simple type of these things is sort of imagining how you can add up sinusoids. So say you have a sinusoid oscillating at a very high frequency, but then you also have one oscillating at a low frequency, then you can sort of get an overlay of smaller squiggles and over a larger squiggle. And this can be decomposed with FFT to show that you have a couple of these frequencies present in the signal. And so on the right-hand side, we can see that there's like 3 spikes approximately that we sort of add up in the time series on the left. So yeah, this concept is pretty tricky if it's the very first time you've ever heard about it. I guess this is like the most general way to sort of understand it is to think about how you can draw a time series signal into a bunch of sums of sinusoids, and then the coefficients of those different frequencies, sinusoids, will become the FFT on the right-hand side that you see. Yeah, so that's a good question. The frequencies, we do get frequencies. So we get like a range of frequencies, and then we get like an amount for each frequency, right? So we have, say like 1 to 30 hertz or something, for example. We might have some low frequency where you have oscillations happening in like the 4 to 7 hertz range, but then you also have things happening in like the alpha range, which is the 7 to 12 range. And so the height of each of these amounts of the frequencies, yeah, usually we have like some sort of power ratio or some one sort of algorithm, which, oh yeah, so power spectral densities here sort of give you this way to more easily compare the two powers of the frequencies. Sorry if that was not clear. It's kind of hard to explain these things. Yeah, let me know if you have any questions about that still. And then second question is the highest spike the only thing that matters for the FFT graph? So if we go back to the previous slide. Oh, Andrew?
Yep.
Could we go back to one slide? Yeah. So here on the right, even though we do see these spikes, these are very simple waveforms. So usually you will not see something like this in the neural data. And the highest spike is not necessarily the only thing that matters, because you could have different waveforms, and then that would represent different outputs of the FFT and different highest spikes and a different second highest spikes and everything like that. So it's not only the highest spike that matters, because if you say say the highest frequency is 10 hertz, and you add a like 4 hertz signal into that, then that signal, that time series signal, is still different from the one that only has a 10 hertz power, right? Even though they have the same amount of 10 hertz power. Yeah, so in this case, you kind of want to basically take into account the entire FFT spectrum, or as much of it as possible. The thing with EEG, though, is that because it goes through the scalp or the skull, it's actually low passed pretty heavily, and so you don't actually get too much outside of like 40 or 50 hertz. It gets very hard to pick up on very high frequencies just because the frequency sort of gets low passed through the skull. So usually, yeah, you would be looking through the lower frequencies. The amplitude for the frequency domain is more of like a magnitude. It has a unit. I think it was like voltage squared divided by hertz or something like that for PSTs. It's like power, so like how big of an amplitude this frequency has. So it's a little bit different from just pure voltage. Yeah, these are great questions. Yeah, and if these don't make too much sense right now because I'm not great at explaining it, then we can always send you guys more materials from videos online. Same for the other domain. Which domain are you talking about? The left side, so time series domain. The S of T down here, the very, very left side. The very, very left side, that would be microvolts. And when we switch into frequency domain, we get something that has something to do with the power, which is usually squared. And then you would divide by the actual frequency to like make the math work out so that comparing equal things. Cool. Okay, so next slide. All right, so power spectral density is a special form of FFT, or it's like a changed form of it. And this one is better than FFT because it does not vary with the number of time points, because you can imagine that if you have a longer time signal versus a short one. So in this competition, you'll be working with 4-second long signals, but we also give you the entire raw data, which is like 40-minute long recording. So you can imagine that if you were to transform that 40-minute long recording into frequency domain versus the 4-second long one, you would get a much different FFT, because the way FFT works is that it's assuming, or it's trying to approximate that tiny signal with sinusoids. And because sinusoids sort of go on forever, you have to sort of squash it in and try to make it fit into that smaller thing. But then if you have a longer signal, then you can have a much larger range of possible frequencies and things that are being taken into account. So with PSD, we can get a slightly cleaner graph. On the right-hand side, you can sort of see how the PSD does not vary as much with the signal length compared to the FFT. Another great thing about PSD is that it has the same energy as the original signal. So yeah, if you were to like integrate it, you would get the same thing as the original signal. So that's pretty nice. Okay, next slide. Okay, another thing we'll be looking at is a spectrogram. And this is sort of like PSDs stacked on each other at each time point. So here on the right-hand side, you can see there's a time axis, there's also a frequency axis. And sort of when we thought about the PSD and the FFT format of the signal, we have spikes at different locations in the frequency domain. And so for every slice in the time domain, we sort of get this FFT. And then when we stack it and push everything through a time series, we can see how the frequencies change over time. So this is very nice for a visual, just like of understanding of the data, but also you could potentially use this as some sort of feature, saying, oh, if at some point then the frequency of something is higher at a certain frequency, then you might give you some idea of what the data is talking about. Yeah, it might make more sense after we go through the data as well. So next slide.
All right.
All right.
So,
go ahead.
all right. So for this interactive track workshop, we would like to have you guys make sure that you have Python 3.6 or later installed. We would also want you guys to have a Jupyter Notebook install, and also the Python libraries installed. So just simply type pip install pandas, pip install numpy, pip install matplotlib, pip install scipy, and pip install neurodsp to install the required libraries for this workshop. And to get the notebooks and data, you just follow the two URLs on the right-hand side of this slide. And they're also posted into the chat for you.
Yeah, so I guess like, is anyone ready to sort of follow along if they can get the notebooks and things cloned? Okay, cool. Okay, awesome. We have a couple people. So the left-hand side was sort of sent through the newsletter. Hopefully people saw that and then set things up. But if you also have that already, then great. It's usually pretty easy to set up Python. The right-hand side, yeah, just git clone that, download the data. Yeah.
Yep.
Okay, so does anyone need more time to get the data and clone it? If you're planning to go through it right now, we will have a session at the end where we'll help you set up everything. But if you want to sort of play around with it while I go through it, then we can wait a little bit.
Yeah, also this session is being recorded, so if you feel like you're falling way too behind and would still like to complete this workshop, you can definitely watch this recording.
Okay, sounds like no one's asking for more time right now, so we can sort of go through the notebook. And yeah, if you guys want to take a look at it after the workshop, we can help you set up as well.
All right, now we're moving on to the next slide.
Yes. All right, so I'm gonna screen share then.
All right, I'll stop the share. All right, Jiling, carry on.
Cool. Okay. All right, so if you were able to go to the GitHub link, you should have been able to see this Jupyter Notebook in it. We also have it on our Kaggle site. So you can see here we have notebooks, and this is the starter notebook. I think you can potentially open this and then run it alongside me as well. It sort of worked for me, but yeah, we didn't really try like someone else trying it yet. So you can also go to this link, I guess. I'll just put in the chat. Yeah. Okay, cool. So that notebook is pretty much the same as this, except with slightly different paths, because in Kaggle, you have like a slightly different path structure than if you were to just unzip the notebook, unzip the data right next to this notebook. So yeah. Cool. All right. Is everyone able to see this? Is anything blocking the screen? Okay, cool. All right, so starter notebook, BCI competition 4, 2B. This is a data set that came from a long, long time ago, in 2008, where a bunch of researchers collected data from people doing a bunch of different tasks, and this one happened to be a motor imagery task. Our data here is a subset of whatever they provided in the original competition. And this data set specifically, they kept in all of the eye artifact data. So it makes it a really realistic data set in a sense, because you can see the eye artifacts and then potentially employ some ways to remove that eye artifact data. In this notebook, we'll go through a single subject's run, and we'll also look at the data in time and frequency domains. Due to the nature of EEG, it's going to look really noisy, so just be prepared for that. And we will be using a lot of EEG feature extraction and machine learning techniques in order to better understand the data. So some terminology, Simon just went through the EEG stuff about like, okay, this is how we collect the data. Basically, someone sticks a piece of metal on your head, and then voltages come out of that. EOG, electroculography, is similar to EEG, but those are on different parts of your face. So then it more picks up on eye movements and blinks. And then PSDs, we just went over that. Spectrograms, we just went over that. Okay, so if you have this notebook, the try sections are good ways to like just play around with the data as well. First thing you'll do is you want to import everything. There's a constant. This constant specifically is just a representation of how quickly the data was recorded at. So this means that we recorded 250 points of data per second. And then we have a bunch of helper functions. The first couple are for data. So getting the data frames from a bunch of things, getting the epoch data frame from the resulting EEG data frames from here. And then also some plotting functions and plotting spectrogram functions. So you can go through those in a little bit more detail to better understand those. But in this part, we'll sort of just take a look at the graphs. Okay, so when you have the data notebook and unzip it, you have a data folder. Inside of it, there's a train folder, which I guess I can share this part. There's a train and test. So train is all the EEG recordings of the subjects and sessions that you will have the labels for, which is in Y-train only. And then test is all the EEG recordings of the people you don't have the Y-train, the Y labels of. So yeah, that's the two differences. And then we have the epoch versions in pickles. Okay, so we basically take the file name and we have these variables that teach us about the different channels just to like label things properly. And then we also have specific event types where event type 0 is left, event type 1 is right. And these are the numbers that you'll see in the Y-train folder and also in the columns that represent trial type in the epoch ones. Okay, so we want to read these in. So we're reading in the raw ones. We see that the recording length is 40 minutes long and we have 120 trials. We have a raw data explanation here. Oh, epoch data. Yeah, it's a good question. I'll get to that after getting through this raw data part. Okay, so raw data is basically we have this huge data frame of time points and the exact voltages at each time point. So in the entire 40 minutes, we have a bunch of milliseconds. This is in milliseconds right here. And because we're sampling at 250 hertz, we basically get one row per recording time. So that's every 4 milliseconds. So every 4 milliseconds, we get a new row. And this gets concatenated. We have a huge data frame. We can see how long it is like this. 60,000 time points. That's a lot. So another thing we want to see in this raw data is that we have this event start column, where if it's 0, it means that's not the start of an event. But if it's 1, it means it's the start of an event. And every start of the event corresponds to the event type, which is provided in the Y-train only folder, where the event data frame tells us that the first row is the event type of the first event start that we saw. What are each of the channels? Yeah, so C3 is, look on the data, if we open this, C3 is this one on the left. So this guy's nose over here. C3 is over here, CZ is over here, C4 is over here. And then on the face, we have EOG 1, 2, and 3. Yeah, this is from, yeah, the 1020 system. Does that clarify? Let me know if you have another question about that. Cool. All right, so now that we know that what these are and all the other ones, yeah, let me know if there's any questions about that. We can try to view this time point by time point. So here we just have some simple plotting functions. This one specifically, we look at the C3, we specifically look at the EOG channel 1, C3, and C3 again. So you can try plotting the other channels. We can also try plotting different time windows over here. So if we currently plot this, this will start at the time point. This is the milliseconds time, so divide by 4 to get the time point. And this is what the data looks like. So in general, you can see how the voltage of the C3 electrode is slightly smaller than the one of the EOG. Usually EOG has a little bit higher voltage just because it's like picking up on more muscle activity, things like that. So that's primarily why. And then over here is a power spectral density of the C3 electrode. So basically this one. We can see how there's this bump at alpha, and this is very prominent in EEG sort of recordings, is that you'll get a pretty large sort of 8 to 12, 13-ish hertz power. And over here you can see in the spectrogram that over time, we can see that later in time we get more power in this alpha band compared to the earlier time points. So yeah, this is kind of like, this hopefully makes more sense in terms of like what a spectrogram is. So like, for example, here at 30 seconds, we get some sort of PSD for some window in the past, and then that sort of gets plotted here. Okay, so epoching the data. This is the part that Schumann asked. And so, yeah, when we want to epoch the data, there's this function called get epoch data frame. And basically it takes in the EEG data, the event data, and the duration for which we want to epoch. Okay, so epoching basically means splitting the data into little chunks of data. So say you have 40 minutes long recording, we want to split it into trials or epochs, where each one is only 4 seconds long. And each one of these 4 seconds longs correspond to the trial that was happening at that same time. So because the experiment had a 4 second long trial session for motor imagery, so people basically sat there thinking about moving their right hand for 4 seconds, we did this as a default, but you can always change this. You can always like try to analyze things in different time frames of this 4 seconds or even outside of it. So yeah, this function sort of provides you the flexibility to change the size of your epoch. Okay, so when we epoch the data, we can see that we get the data in terms of trials. So each row, instead of being every 4 milliseconds of data, we get 1 trial. So each one of these is 4 seconds of data. The start time is basically when the trial started since the beginning of the recording. And then event type, again, is like left or right. So in this case, this would be a left trial. And these are all the data that are associated with it. Yeah, so pretty simple, right? Yeah, so that's one way you can get the epochs. Another way you can get the epochs is by loading in the epoch train pickles. These are basically data frames that we pre-epoched already into 400 milliseconds. And so you can load all of these and they have their patient IDs and the start times given to you already. So you can sort of analyze it by patient ID and also like just have all the data linked together. So this is good for evaluation too, because we have we'll have something in this form for you to input into your system for evaluation, and then your system will have to output predictions for the event type. So that's why these were provided. The test one that I sort of mentioned before, epoch test, that one's kind of like the input to the evaluation, and then your output should be your predictions. Okay, cool. So yeah, any questions on that so far? This is basically getting the data, viewing the data, and how the data sort of relates to the competition. A lot of cool things you can do to like explore. Like one thing I could do, this might be fun, is sort of find an egg link. Let's see this one. There are other ways to clean the data that are sometimes used but not shown here. Yeah, so here I show you the EOG and the EEG channels. One common way is to use ICA, independent component analysis, in order to extract the eye movements from the EEG data. So that's commonly used to make sure that we don't confound our EEG data with eye blinks and eye movements, eye surprise, and things like that. So yeah, there's definitely other things. You can bandpass filter these. These were previously bandpass filtered by the competition. So originally, they already took out the power line noise. They already chopped it so that like the lowest frequencies and the super higher frequencies, like above 50 hertz or so, are basically eliminated. So you're already sort of working with slightly clean data. Usually you would have like a much more trendy sort of EEG data that sort of like goes down or goes up. Yeah, cool. Yeah, we'll definitely learn more about other ways to look at this EEG data in future workshops. Yeah, I'm trying to find an iBlink one. I guess that wasn't the one that had it. If I don't find it soon, we'll just move on for now. But yeah, you can try exploring this and then seeing if you can find the one with the iBlink. It might take a few trials. Maybe I'll make it like a longer signal too. We can see.
Jiling, we got another question just popped up. So somebody asked, can you explain the difference between the data folders again via test, train, etc.?
Yeah. So test and train are both the raw recordings, kind of. You can sort of think about it that way. It's been through a lot of processing already, but they're basically really long files of recordings of EEG data. And so the train one specifically is the one that has the labels given to you. That's why we separate into two folders. The test ones, we didn't give you the labels, so that's why they're in a separate folder. Yeah, so the test and train ones are the ones that get loaded into these data frames. And as we saw here, we can sort of time point window these things and then view them over here. Hopefully that answers that question. And then in essence, all this data is, yeah, we have a device connected to someone and we read the voltages that the brain outputs, yes, and we can derive what region of the brain is working. So yeah, region is one thing you can derive. Source localization is one thing that people do with EEG data, because you have multiple locations you're recording from, right? And so you can sort of mathematically approximate where certain signals might come from. Though this backward process is definitely not a very robust one, because signals from different locations in the brain can make it look like it's coming from another location, right? So you might hypothesize that it's coming from your hippocampus, for example, but then it's actually coming from somewhere else. So people do it, but it is one thing that people do. But it's like a little bit more difficult to do, especially if you only have 3 channels here in this case. Other things that we can derive from this data is, for example, like when people are blinking, looking at the frequency domain, we can try to extract information about people's focus levels, maybe if they're sleepy right now. Yeah, in this case, we want to sort of extract if people are thinking, moving their left hand versus the right hand. So it's not just region. We can try to find other things too. Cool. Output data. Yes, labels. Yeah, labels are in the Y folder. And also if you use the epoch ones. The epoch ones, I'll show you right now. Did I not go through this yet? Yeah, epoch ones have this event type that have the, basically it's 0, 1. So 0 is left and then 1's right. And if you load it in basically like this, then you can read it and see. Cool. Yeah, so that's basically how to get the data, what the data means, and what you might want to do with prediction. Prediction is not the only thing you might want to do with this, because it's because if you might be starting out with like neural data, another good thing you can do is sort of just visualizing it and understanding it in different ways. So doing like the frequency domain analysis and trying to come up with creative ways to visualize the data is also another approach. to the competition. Cool. Yeah, good questions. Okay, so sort of going more in depth, if we have this epoch pickle, we basically pickle holds like some pandas data frame information in it. So if you load it, then you get a data frame. By looking at that, we can use these plotting functions to look at the data at each trial number. So in this case, we're going to look at trial number 0. This is what trial number 0 looks like for all 3 channels. It's kind of hard to analyze if you look at all 3 channels at once, but I provide you the code to do whatever you want with it. But here at the bottom, we can sort of see some interesting things happening with the power spectral densities. We can see that here in this case, we see a lot more alpha power in the C3 electrode than the C4 electrode. And the CZ electrode kind of just does whatever. It's like the center electrode. So usually when you look at trial by trial, it's kind of hard to read the PSD because there's just so much noise, as you can see. There's so much like power in this like higher frequency and also this lower frequency squiggles that it doesn't really mean too much. But this one's a kind of a good example of like what might happen in each trial. A better way to understand it, especially in the cognitive neuroscience domain, is to sort of average everything and then just see like what the actual like, outstanding differences are. So if we took all of this data and we took all of their PSDs, and then for every 4-second interval, we got this PSD for each trial, we averaged everything for the left and the right trials independently, then we can get the average PSDs here. And interestingly enough, as we learned from Simon, this we can see how when someone is thinking left, that the C4 electrode has slightly less alpha power. So again, C4 is the one that is on the right-hand side, the contralateral side. This is the phenomenon of the event related desynchronization that happens when you try to move the contralateral side's hand, then the other side's alpha power sort of goes down. And similarly, we can see this on the right-hand side thinking processes. So when someone's thinking right, on average, the C3 electrode is slightly smaller in power than the C4 electrode. C4, yeah, C3 is the left-hand hemisphere, and then you're thinking about moving your right-hand side, so it kind of goes down. So this is sort of the cognitive neuroscience finding that this sort of event happens. And it might be like one feature that we might want to use for distinguishing the 2 classes in real time. But because, as we saw with such noise and how like each trial's PSD looks kind of crazy, there's no like, usually just putting a threshold is not like enough to see if like, oh, if the C3 power is at 10 Hertz is above a certain threshold, we might think it's left. We can start with that, but there's always a lot more things that we might want to do to improve our models. Cool, yeah, so that's pretty much the introduction to it. There's some EEG, oh, yeah, these stats are kind of interesting too. Yeah, I'll just go through these really quick. If we take the mean voltage of the EEG channels, we can see that the average is approximately 0. This is because we band passed everything, so the low frequencies that cause any DC offset from the center mean will flatten the entire signal so that you have everything around 0, basically. And then you have the standard deviation here. This thing tells you like how much it fluctuates from like the average. And so we can see how it fluctuates actually quite a bit, a few volts, a few microvolts, I mean, sorry. And then the range is actually pretty large. Oh, I did not run it. It's about 150 microvolts or so, as you saw before. So the lowest one, the highest one minus the lowest one is pretty large range. within a session. So yeah, and then down here you can get some more stats of maybe the EOG data to see what that looks like and get some practice with doing these statistics. You can also find other statistical methods to analyze the data. Cool. Anyone have any questions about the notebook? What are some ways that one might want to explore this data? What kind of things might we want to make statistical inferences about? Right, so some ways we might want to explore this data is we could explore it visually by looking at different time windows. We can also look at it in the frequency domain, in spectrograms. We can also become more creative with how we time window these things and find the PSDs of those different time windows. Other things we can do is like we could look at the differences between 2 time points and see what that averages. Yeah, there's a lot of things, and we'll go through more of them definitely in the EG feature extraction workshops. And some other things we might want to make statistical inferences about. Yeah, as I mentioned, the difference between time points, that could be something you might want to look at. Looking at the different standard deviations of the power spectrums, maybe, that could be interesting. Is the 4-second window in each trial strictly the motor imagery phase? Yes. Yeah, so I guess another good resource I'll just show right now is this PDF. This is like the original PDF that they gave people on like what's in this data set. There is a diagram of this part. So there's this imagery period here. Cool. 1, 2. Oh, is it? Oh, maybe it's from starting from here. 1, 2, 3, 4. Okay. Spectrogram, little dashes of color, the spike in frequency domain, yeah. So the yellow should specify a higher power in that frequency. So in this case, we have like some like delta power at like times second 5-ish. Cool. I guess that's most of the notebook walkthrough. Yeah. Oh. Higher frequency color mean a higher spike in the frequency domain. Yeah. So if we were to look at this, if we were to have this dash of color slightly above here, so like in the 25 Hertz range, then we would expect to see at that time point, that window would have some spike like over here. Can you see my mouse, by the way? I'm not sure if I'm just waving in there. Okay, cool. Yeah, so it would be like over here. That makes sense? Cool. Yeah. So yeah, that's pretty much most of the material that we had planned for today. We have some time, so we'll just go through, these are the upcoming events that we're planning to have. In week 5, feature extraction 1, so we can continue learning about neural data. We'll have destructured social to get to know each other a little bit better in week 6, and then feature extraction 2, and then a competition. So yeah, we're doing a motor imagery competition on the Neurotech X annual competition level. And so we'll present that in week 9 and have a social there. In the winter quarter, we'll wrap up the competition, and yeah, be sure to check out the Kaggle competition page to see what the details are, the rules, and also what the prizes are. Well, we have to specify that still, but there'll be, as Darren mentioned, gift cards. And then, yeah, check out our Facebook page. Okay, so yeah, let's just move into the open hours for getting notebooks working and answering any more questions you guys have for getting started.